hello, and thank you very much for inviting me to this very historic conference. It's a great honor to be here. And although you must be thinking, what the hell has this got to do with anything? You might find that there are some interesting parallels with pediatric work. And just listening to the conversations, I can already see that we can learn a lot from each other. We know that there's a rising incidence of cancer. You probably don't need me to tell you. But it is quite scary when you think that by 2070, there are estimated to be 34 million cancer cases diagnosed worldwide. That's quite a shocking statistic. And that actually, at the moment, cardiovascular disease is a leading cause of death, but in the future, it's going to be cancer. And that means for our children's generation as well. We talk about this as the silver tsunami. And are our healthcare systems ready for this? And the answer is definitely not. So as a result of that, there's been a lot of pressure, a lot of guidance around, I'm sure you are aware, about ways of preventing cancer, dietary modifications, lifestyle choices. But the truth is that although that does prevent 40% of cancers, that mostly benefits those living in countries with a low human development index, low HDI countries, where there are endemic problems with smoking and pollution. But if you come from countries with very high HDI, like in Europe and Northern America, actually you need more than just a passive form of prevention. You actually need to think about active forms of prevention. And by active, I mean drugs or vaccines. And this is the NHS long-term plan. There's a lot of talk, this is a previous one, there's a lot of talk also from the NCI about prioritizing preventing cancer. And CRUK has a very ambitious plan by 2034, so we're looking at in 10 years' time, that they want to accelerate progress so that 3 out of 4 people survive cancer. At the moment, 50% of people with a cancer diagnosis live beyond 5 years, which actually is quite shockingly bad when you think about it. So we've got a lot of work to do and only 10 years to do it. And if we go with existing trial designs, and I'll talk to you about that in a minute, it's really impossible to do that. But what surprises me, and actually even surprises oncologists, is that cancer does not appear out of nowhere. Cancer takes a long time to develop in our bodies. And so if you look at something, for example, like liver cancer, it takes 10 to 20 years after a diagnosis of, or after something like an infection with hepatitis C, for example. Ovarian cancer, 4 to 6 years. 4 to 6 years. It doesn't even start in the ovary, it starts in the fallopian tubes. So it's a complete misnomer. We're missing it. Colorectal cancer, we're fortunate that we can see polyps. But again, 5 to 15 years, but that's even conservative. It's probably more like 20 years it takes to grow in our bodies. 20 years. Squamous cell lung cancer, 8 years. And so if we want to prevent cancer, we first of all need to be able to recognize where a cancer is growing in our bodies. And at the moment, growing cancers are pretty much invisible. You can't see them on CT scans. You can't pick them up with blood tests. The only ones that you can actually detect are colorectal cancer, because you can see the polyps when you do a colonoscopy, and similarly cervical cancer. And interestingly, if you think about it, those are the 2 cancers where you've really seen the benefit of screening, because not only have they screened for that, but they've also resected it at the same time. So it's actually a preventative intervention. So how can we change things? Well, first of all, we need to look at people with at risk of cancers to guide us, because we're not really using our at-risk community as much as we should be. So if you have cirrhosis of the liver, you are at very heightened risk of getting hepatocellular carcinoma. And these patients are coming into clinics and having ultrasounds, but we're not doing anything. We're not giving them any interventions. If you have, I'm sorry, I'm finding it hard to press all these buttons. Smokers, for example, squamous cell lung cancer, their risk is about 10% during their lifetime, but there are certain peaks during the disease process, which I'll talk about in a minute, that actually you could intervene and prevent them from getting cancer. If you then, for example, ovarian cancer, there are patients or people who have BRCA syndrome. They inherit a mutation to BRCA1 or BRCA2 gene, and they have a 50% lifetime risk of getting ovarian cancer and an even higher risk of breast cancer. What are we doing in that group? Are we trying to find precancers? The answer is, at the moment, not really. And then if you've got if you have Lynch syndrome, you have about a 70 to 80% risk of developing colorectal cancer during your lifetime. So we are starting to think differently about cancer and starting to purposefully look in these high-risk groups to help us understand what's going on. And so I talked to you about these latent cancers growing in our bodies, and I want you to learn a new word, which is pre-cancer, and that's what we're talking about. So by the time we get to see a patient in clinic, we're looking at the above the waterline, a diagnosis of cancer, but we are missing this huge, often decades-long stage when it's in the pre-cancer stage. And actually, what happens during that stage? What happens during pre-cancer? And intriguingly, we don't know the answer. So you think, well, cancer's complicated. We've been talking a lot about monogenic diseases in childhood, but actually cancer starts off as a single mutation. It starts off as a monogenic condition, a mutation in often a single gene, and then it snowballs and becomes very, very polygenic. And so it's almost impossible to look at things like antisense oligonucleotides in cancer because you're really dealing with a very heterogeneous and genetically messy disease by the time it becomes diagnosed at this end. But what we really need to do is we need to shift the dial and start trying to understand what happens at these earlier stages. We know that there's dysplasia, we know there's inflammation. We're starting to get an idea of the biology, and that's something we're really focusing on in Oxford, trying to understand pre-cancer biology. And so where can we intervene? Can we actually intervene before a cancer becomes diagnosed? Can we look at using a vaccine, for example, in the years before a cancer diagnosis? Particularly if it's a decade long, we could potentially go in this high-risk population, knowing they're probably statistically harboring a pre-cancer at that time, and test an intervention. Because if you otherwise go with the standard population-based prevention design, these studies generally, they recruit tens of thousands of people. They yet tend to use a repurposed drug like aspirin or something like that in this great big population. The studies are too large to do any biological readouts. So you end up with a very, very long study. It reads out after about 10 years because they're looking for overall survival as the outcome. And at the end of it, you're left with more questions often than answers. And pharma are not attracted to running these kind of studies. They tend to be academically funded trials. So if we're going to run these studies in this high-risk group, we're going to have to rethink the way we run the studies. And that's, again, something we've been focusing on. And we now are calling these precision prevention trials. So instead of going with a population of risk unknown, we are focusing entirely on the high-risk group of patients, many of which were excluded because of their syndromes from other studies because their risk was too aberrantly high. So this is the focus of precision prevention trials. And I put it back to you that actually, in answer to some of your N=1 questions, you should be looking at biological endpoints because you can design trials around that. And that means that the burden is not then on endpoints around overall survival. So we're looking at these studies, I say small, we're talking up to 500 patients, genetically high-risk groups on the whole, but they can also be environmental high risk. But they have to have biology that's matched to an intervention. We have to be able to have a scientific hypothesis and a matched drug. And then we will do serial sampling of the patients to try and understand that pathway to cancer, which we still don't really understand. And so that we can develop those surrogate biomarkers. It's a problem for us too. We just don't have surrogate biomarkers. And benefit the group itself, because they're a high-risk group for which very few drugs are ever approved because their numbers are too small to run big studies. But also for us, it gives us enough evidence to learn from them to help identify patients in the more population-based approach, but at the next stage of our studies. So we were very lucky. We applied for Oxford to be the UK's first centre dedicated to precision prevention trials in the UK, and we were awarded CRUK funding. And so we are now in the first year of this funding, and really now starting to establish the collaborations and starting the work that we really proposed when we applied to CRUK. The purpose of us is not to run these studies. The purpose of the studies is to be run elsewhere, but for us to really work on the infrastructure and the design of the trials in Oxford. And so OCTO, which is the trials unit that I lead, we have on our tablets of stone sort of 10 main components to our studies. And we try and make sure that this kind of toolkit is embedded into every study that we do. An important component is really embedding the voice of the patients and the public. And that's something that's really come out strongly at this conference, and that we've also learned that you can't make decisions on behalf of patients or public or high-risk groups. You can't make decisions about risk versus benefit without engaging with them. And the answers will surprise you. We want to be able to have biology at the core of these studies, be able to build trials on trials, be able to recruit patients from far and wide, so make the studies easy for people to be enrolled into, and share our practice with others. So you're thinking, okay, what high-risk groups can we go into? There aren't that many. The first and most obvious to me is Li-Fraumeni syndrome. There's about 700 people living with this syndrome in the UK. They have a genetic mutation to P53, which is a tumor suppressor gene. So without it, their risk of cancer goes up to 90 to 100% during their lifetime. It's a guarantee, basically, of getting cancer. And so the first study that we've designed is the Mili study, which is at the moment being run in this group. Also, we've got a study in Lynch syndrome looking at a vaccine, and we're also developing a study called BRCA-vax in breast. And in addition, we're also running another study, about to start running another study called LungVax. And I'm just going to very briefly just touch on this, just to give you the principles. But the main questions that we ask are three. Does the intervention prevent or delay cancer? Of course, that's the really number one question that the patients and we want to know. But we want to also know, how does it prevent cancer? What are the molecular events leading to cancer? We still can't answer those questions. So the Mili study, for example, is based on some work that came out of Paul Huang's lab, who's now at ICR. And he discovered that people with a P53 mutation have aberrantly high oxidative phosphorylation in their mitochondria. In other words, their mitochondria, which is like the cellular boiler, is turned up too high. And so they can't turn it down, and the fact that the boiler is turned up too high is inducing a higher risk of cancer, mechanism not quite still known. He demonstrated that you could use metformin, which we all know to be a very common diabetic drug, and that actually one of the roles of metformin, it has multiple functions, but one of it is that it turns down that boiler. It has a direct effect on the electron transport chain in the mitochondria. So he did that. They did a pilot study, demonstrated that was true in the States, and then we designed a clinical trial using metformin in patients. And this is the sort of basic design of the Mili study. Patients are randomized 1-to-1 with Li-Fraumeni, anyone with a confirmed Li-Fraumeni syndrome, and they receive metformin daily for up to 5 years versus a no treatment control. We had a lot of interesting discussions with patient groups about that. And their opinion was, interestingly, because it's a hereditary condition, autosomal dominant, they often see their parents, brothers, and sisters die of cancer during their lifetimes. So they have a strong feeling of wanting to give forward. And so for us, we didn't have the problem that you have in the sort of pediatric setting of aversion to placebo. But we do have a deal with the patients that they'll tell us what they're on. You know, they can take other things like aspirin, et cetera, to reduce their cancer risk. And so we've started recruiting to this study already, and we are very strongly reliant on our patient group, advocate group, who are helping us along the way in publicizing the trial. And from this, we want to be able to answer questions about truly how does metformin really work? And also we want to understand, how does cancer start in these people? And then really, does metformin work in this group? And so to answer the how does it work, we're working at sort of integrating data that looks at different aspects of the biology and converging it together to understand how metformin works. Does it directly work on that thing I told you about, the oxidative phosphorylation aspect, or is it an indirect effect on other aspects such as levels of insulin and sugar and growth factors, which are all known to stimulate cancer? The likely answer is it's probably a bit of both. And this guy, Andy Blake, who was mentioned in the previous slides, is very involved through this data translational platform. So this means we can get data back immediately as we're running a study. And this is something that pharma companies cannot do, but we can, because we have the links to be able to link all the patient data, all the incoming RNA-seq and proteomics data, and basically crunch it into a C-Bio portal so we can see in real time what's going on with our patients. And this is my understanding of what that means. So if we're looking at the different aspects of metformin, we're feeding in these different factors and then comparing it with clinical outcome. And this is really the group heavily involved in this, but particularly the PPI group, which has been absolutely amazing, the public and patient representatives. And so for us, what's been really lovely about this is we've been sharing the protocol. So there are now international Mili studies going on. And the idea is we're making sure that we all synchronize this so that we pool, in the end, the data together and get some really nice understanding of what's going on in these patients in a larger meta-analysis, because it's a rare cancer. It's very hard for individual studies to answer a lot of questions. But the real aim was Mili-Ped, which is the pediatric version. And I believe, well, there are some pediatric centers who are thinking now in the UK about running this study with liquid metformin in younger children. I'm very keen to see that happen. I think it's lovely if we can bridge pediatric and adult studies, because I should say in Li-Fraumeni, there's a peak of cancer at about the age of 5, and then there's an adult peak at the age of 30. So we're definitely chasing the adult peak, but what about that pediatric peak? Wouldn't it be nice to look at that? I just want to very quickly tell you that LungVax has taught us a lot. And this is something where we've discovered that pre-cancer cells, even smokers, long-term smokers, before they even get cancer, they start expressing a signature of neoantigens expressed by MHC molecules, HLA molecules on their cells. And that signature continues all the way through to them developing cancer. So we're developing a vaccine to target that signature in smokers. And this work has really come from some fantastic studies where they serially sequence the tissue from smokers before they developed cancer and then onwards afterwards. And we've been working with UCL around this and working with the Tracer X team, which is a very large group. But we're chasing a peak in this track because you have to really, with these precision prevention studies, you have to stop them from becoming massive population prevention trials. We have to chase a peak in incidence. And in this case, there is, for patients who've had an early stage cancer resected, there is a heightened risk, not only of recurrence, but also of new cancers in that group in the first 2 years since their surgery. And so we are really focusing LungVax in that group, and we've got our interview with CRUK tomorrow as to whether or not we've got funding. And the reason that they also get other cancers is because the whole field is also affected by the long-term smoking. And once you treat one cancer, it's a bit like whack-a-mole, other cancers appear. And so for us, it's going to be really interesting. We've designed a vaccine that's targeting these mutational hotspots that appear and are quite common across these cancers. So it'll be really interesting to see the impact of this vaccine on other on those other cancers. And this, again, is a similar design. We're doing a safety run and then a randomized study. Again, we're using a no treatment control arm with the patients wanting that. But I suspect we may end up having to, in the end, under pressure, go for a placebo control. But we're going to be asking similar questions, biological questions. If we see that the vaccine raises an immune response, does that actually correlate with them not getting cancer? We can't make that assumption that it will. So this is an important endpoint for our trial. And it's really nice working with people like Dame Sarah Gilbert, because we're using the basis of the ChAdOx for the vaccine. And people like Charlie Swanton, who's very well famous for his work in this field. So the nice thing about these studies is they're very, very collaborative. We've worked with patient groups extensively and done a lot of town hall meetings to find out what they want. And the message loud and clear is don't make a decision about us without us, which is something we are really kind of integrating into our work. And BRCA-vax is another, which I won't go into too much, but just to say that at the moment, the option is surgery. Remove your ovaries and fallopian tubes. A very, I fear, masculine solution to the problem. What we would like to do is to develop a vaccine in this group as well. But ultimately, our goal, our real overarching vision, is to have a series of these studies. LungVax is due to start in January 26, with funding, hopefully this week, approved. LynchVax is the next one. I didn't talk about that, but we've developed a vaccine against lynch. EBV is another one. And we have a list of these vaccine studies. And we're discovering that there are similar epitopes. So we're seeing that vaccines aren't very different to each other. So our ultimate goal is really to think about running AllVax, a study where we have a master protocol. It goes back to the problems we were talking about with the jammy dodgers, but have a master protocol that we can cut and paste these vaccine studies, and we can collectively pool the cancer incidence as we go and get a sort of so we compare it overall. Because what we'd like to do in the end is pool the vaccine and have one vaccine that we're going to give to everyone, if they want it, aged about 40 years old, to protect them from the 4 main cancers. And that is our goal, and we want to achieve that within 10 years. And we're calling that the Cancer Shield vaccine. So our ambitions really are to use the high-risk group to understand the benefit of the vaccines and then extrapolate that to a more general population, so that in the future, cancer will be a thing of the past. So I've told you that people with cancer susceptibility syndromes are often excluded from studies because of their untypically high risk, but often the biology is similar, so we'd like to really start running our studies around them. We want to target vulnerabilities with precision prevention trials. There needs to be biology behind it. We need to discover surrogate markers so that we can attract not only better studies, so we don't just have to go into high-risk groups, we can identify through a blood test that someone's at risk of cancer or someone is brewing a cancer. And for us, that also attracts in pharma because you can run it with a shorter time scale and it's a lot more attractive. We have a pipeline of drug and vaccine precision prevention studies. I've shown you that. We're running them nationally and integrating the patient and public in this. But we feel ultimately these epitopes could be pooled into a single cancer vaccine. Because if you start thinking about risk, at the age of 50, you have now a 1 in 2, if you're a male, chance of getting cancer before you're 70. And if you're a female, it's 1 in 3. And I think that is pretty high risk. So age is ultimately another cancer susceptibility syndrome. So thank you.